Hello everyone and welcome. My name is Sarah Kirkpatrick and I'm going to be talking about how vegetation communities and pore water nutrient concentrations are different where Typha x glauca and shrubs are prevalent in a coastal peatland on Lake Ontario. My major advisor is Dr. Rachel Schultz and my committee members are Dr. Michael Chislock and Andy Graham. Great Lakes coastal wetlands have gained a lot of focus in wetland restoration and conservation initiatives. One of the many reasons behind this is that they serve as the kidneys for the world's largest freshwater system. They filter nutrients, pollutants, and sediments. They also mitigate the impacts of floods and drought. They also are a unique ecosystem for wildlife and vegetation. Peatlands are a type of wetland that can be found along the coast of the Great Lakes. They have unique environmental conditions that support uncommon or rare species. They also store large amounts of carbon, making them the largest terrestrial carbon store in the world. Because of these ecosystem services and their widespread degradation, peatlands are often targeted for conservation and restoration. One threat to peatlands is ecosystem change. A driver of this change that is seen throughout Lake Ontario wetlands is the invasive species Typha x glauca, or hybrid cattail. Another driver of change in peatlands is the encroachment of woody species such as alder. Both of these drivers can alter nutrient availability, hydrology, and light penetration. Because peatland vegetation grows in low nutrient, inundated, open habitats, invasive cattail and shrub encroachment can be detrimental to vegetation communities that are specifically adapted to these conditions. This may also reduce the floristic quality of a peatland. The questions I aimed to answer were 1. Is there a relationship between floristic quality and shrub or cattail cover? 2. Are vegetation communities different in areas with greater cattail or shrub cover? And 3. How do nutrient concentrations differ in areas with greater cattail or shrub cover? To answer these questions, I selected my study site, which is the coastal peatland on Lake Ontario, being invaded by Typha x glauca and encroached upon by shrubs. I also selected a reference site in pristine condition to compare my site to. My study site is located at Cranberry Pond, which is part of the Braddock Bay Wildlife Management Area. My reference wetland is located at Lakeshore Marshes Wildlife Management Area. Both of these locations can be seen in the map below. I randomly selected 6 sites, each with varying levels of invasion of cattail. These invasion levels are depicted below: a less invaded area on the left and an intermediately invaded area on the right. Each invasion level had 6 3-meter by 3-meter plots. I also installed 4 groundwater wells called piezometers to establish a foundational understanding of the groundwater hydrology of my study site. In Figure 3, you can see the coastal peatland area outlined in green. My study site locations are blue and the piezometer locations are red. For my groundwater hydrology study, I collected groundwater samples seasonally to determine alkalinity, specific conductivity, pH, and temperature. I also measured groundwater levels seasonally in a nest of piezometers located at the center of the peatland. This will help to determine the direction of vertical flow. In each of my plots, I estimated percent cover of plant species and detritus to calculate vegetation metrics, including weighted mean C and cover-weighted floristic quality, using the equations below. I also collected water from the peat, which is called porewater, and analyzed these samples to determine nitrate, nitrite, orthophosphate, and ammonia concentrations. So returning back to my questions, the first one was if there is a relationship between floristic quality and cattail or shrub cover. In Figure 4, we can see a strong negative relationship between cattail cover and floristic quality. We also surprisingly see a positive relationship between shrub cover and floristic quality in Figure 5. Restoration efforts to remove cattail or reduce its cover may improve the floristic quality of the peatland, which is a common goal for land managers. My second question was, are vegetation communities different in areas with greater cattail or shrub cover? As you can see in Figure 6, Typha x glauca was associated with vegetation that had a lower affinity to high-quality habitats. Shrubs such as alder and morella were associated with vegetation that had a high affinity to high-quality areas and peatland-specific vegetation. You can also see that the reference site had different vegetation communities than that of our study site. My third question was, how do nutrient concentrations differ in areas with greater cattail or shrub cover? Plots associated with greater cattail cover also generally had higher nutrient concentrations, as seen in Figure 7. However, nutrient concentrations were found to be within reference levels, as seen in the table above. Nutrients were also generally lower in areas with greater shrub cover. There may be many reasons behind this to explore. Using the groundwater level measurements taken from the piezometer nest in the center of the peatland, we were able to determine that there was discharge groundwater flow in the spring. These effects disappeared in the summer and fall, possibly due to higher evapotranspiration rates. As seen in Figure 9, alkalinity was highest in the western piezometer, corresponding to calcium carbonate deposits, as seen in Figure 8 in blue. Specific conductivity was highest in the south and the center. Understanding the influence of groundwater can help to explain patterns in vegetation communities. Moving forward, we can use these observed patterns to inform management decisions. Last summer, several restoration techniques to control cattail and shrubs were implemented in an effort to improve floristic quality and support peatland vegetation. This coming field season, we will observe any changes in vegetation metrics such as floristic quality and changes in porewater nutrient concentrations. Successful techniques may be applied to restore this peatland and any other degraded peatlands. Thank you for joining me today in my talk, and please feel free to leave any comments for questions. Thank you.